So the tutorial that we are going to run, you also got instruction for it, but you want to access it on your browser as well so that you can have all the instructions next to your terminal. So if you need to find it, there's a link in the instructions, but if you want to find where it is, I'm going to put it as well in a chat window of Zoom. So you should go to bonvinlab.org, which is the group page of my group. And once you are there, you should go to Education, where you find a list of tutorials. They're also recording on some of our lectures. So scroll down and then you find tutorials for HADDOCK version 3. That's where you want to go. And here we have different ones. You also find glycan modeling, for example, basic DNA. But for today, we're going to the antibody-antigen docking tutorial. So let me introduce the tutorial, and then you can go to work. And the plan is I will catch up from time to time to at least demonstrate a few things if you're lost, so that we are at different stages. But in principle, you should be able to follow things on your own. So the entire tutorial is here online. It's always available. There are different setup. If you look at it, for example, there is this link here, BioExcel HPC workflow, workshop, Sofia. So this is what you will need to do on Discoverer to copy the file that are needed to run things. So let me just guide you first what we are going to do. So we're going to model the interaction of an antibody with its antigen. So this is a full structure. Let me change. So now you should see better my mouse. So this is a full structure of an antibody, but what we are going to concentrate on is only one of the arms. So an antibody has two binding domains. So we're going to model this region binding to its antigen. And we're going to use the information of those hypervariable loops in the antibodies to guide the modeling process. So the antibody that we're going to model is called Jevotizumab. It's actually a monoclonal antibody, and it's used against interleukin-1B. So it's a drug, actually. So it's a therapeutic drug. And for this system, we have NMR data. So there are NMR studies that have been done that are defining the binding site on interleukin-1B. So we have information on the binding site for the target, the antigen, and we have the knowledge about the CDR loops for the antibody. And that's the information that we're going to give to HADDOCK. So now all the tutorials that you find on our webpage have this color coding structure. So orange is a question, so you have to think about it, okay? Blue is an instruction. You will have to do something. Green is for visualization. So in this tutorial, green will be PyMol commands. You cannot visualize from Discoverer. So if you want to visualize file, you will have to copy the files back to the Linux desktop and visualize there. But visualization is nice, but it's not critical. So visualization, I might do it from my laptop just to visualize, to show you what to expect. And if you don't manage to do it, it's no big deal. And black is a Linux command prompt. So these are commands that you will have to give in a Linux terminal. So this means in this particular case on Discoverer, okay? So in principle, you can copy-paste commands. So this is a general concept. We have other tutorial where you will not see any black, because then you are using, for example, the web server of HADDOCK. So I don't need to explain you how HADDOCK is working. So here we are going to use HADDOCK3, which is available on the Discoverer, which has been installed. And here are already the first instructions. So if you are logged in on Discoverer, so you should not do this part. You know, if you do things on your own laptop, you can run this tutorial, but you need to have HADDOCK installed. On Discoverer, basically, you should follow the instruction, create a directory as indicated with your username. And once you are in that directory with your own username, you want to basically give these commands here, which is unzipping. So there is on the file system, there is already an archive that contains all the data for the tutorial. And these archives also contain a script to submit to the Slurm system of Discoverer. If you download the data from the web, which is the first instruction, it does not contain the discover specific script. It's just a generic, so you will have to write that script yourself. But the data here are on Discover, so you don't need to go over the internet to unzip them. And after unzipping, you will get this HADDOCK3 antibody-antigen. In order to be able to use HADDOCK, of course, you should start with srun a connection to a node. So this was given in the separate instruction that was sent to you. And then to activate HADDOCK on Discover, you need to load those two modules. So a Python 3.12 module, so HADDOCK supports Python 3.9.12. And then you load the HADDOCK3 modules. And this is defining then all the software that you need for this tutorial. So the local setup instruction was if you were to run things on your own system. So what are we going to do? So once you are on Discover, you have enabled the HADDOCK module, you have unzipped the data. There are different things that we're going to do in this tutorial. So I'm just going to show you very quickly what are the steps. And then you can go on your own. And then from time to time, I will catch up. So there's a first section about preparing the files for docking. So here we're going to download the PDB files from the database, from the protein database, and we're going to pre-process those files, for example, to remove water and remove any crystallization molecule. You know, it's never a good idea to take a file from the PDB and use it blindly because it contains a lot of junk. So this is what it's about. The other thing about antibodies and HADDOCK is that antibodies consist typically of two chains. So there's a light chain and a heavy chain. And in the PDB files that you download, they will be labeled as chain H for heavy and L for light. And the numbering of residues in those chains might be overlapping. In HADDOCK, one molecule is one chain. So we need to renumber the antibody so that we provide the heavy and light chain as one molecule, but they should not overlap in their numbering. So we are shifting the numbering of the light chain for that. So this is what is described in preparing the antibody structure. And we use for that PDB tools, which is a set of tools that we have developed to basically handle PDB files. The same for the antigen. Then we need to define the restraints to guide the docking process. So we're going to define the paratope, that's the binding site on the antibody, identify the epitope, this is based on NMR data, and generate the restraints. Once we have all of that, we are ready to run. Then we have to define a workflow for HADDOCK. So they are, and then we have to execute that workflow. And the execution on Discover is described here. There's actually, when you unzip the file, you will find in the directory HADDOCK3 antibody-antigen, there's already a script which is ready to be submitted to Discover. So actually, you could directly submit that one before starting to do anything because all the data that you need are already provided. You want to go for the step just to learn to do the steps, but you can already run, submit to Slurm that script so that you get your data within 10 minutes or something, 10, 15 minutes, depending on the availability, okay? Once things have been running, we can start analyzing the results, look at the statistics, looking at the scores, looking at the models. So this has some components that do, for example, use PyMol. So maybe I will describe that. I will show you that for my laptop. Otherwise, you have to transfer the data to your desktop system if you want to do it yourself. And at the end, you know, if you are very fast and you are a pro user of HADDOCK and you are bored, there's a number of bonus sections you can run. If you are ready to go, you have your connection. The first step that you will need is to go to this setup for BioExcel on Discover. You got the instruction to create a directory. You should use srun to start a session on Discover. This is where we're going to work interactively. And then you can start following these instructions. So you don't need to do the local setup because this is if you were to do that on your own laptop or on your own. So, and then the first step will be to prepare the files for docking, okay? So that's the first step to go, preparing files for docking. So if everything goes right, if you are connected, you start a test run, you unzip the file, you create a directory, unzip the file, you should see something where you are in this directory and this is the content of the directory. Okay, if everything is there, then you should see this. So I'm actually logged in on Discover now. So I can run it on my laptop, I can run it here, it doesn't matter. If you unpack the data, this is what you are seeing, a directory where there is a PDB directory, restraints. This file is specific to the zip archive that you download from, that you unzip from Discover, because this is a batch script for submitting to the Slurm, the system. There's a directory run, which contains pre-calculated data, some scripts for analysis, and a workflow. So we can just look at, just look at the content. So this PDB directory contains different files. Some of those are models generated with AlphaFold or Immune Builder. These are AI models. This is related to the bonus sections. There you see the reference structure. This is what we are trying to rebuild. And these clean files are the ones that are coming out of the PDB tool processing that we are going to do, which is described. But in principle, everything is pre-calculated and there. The other files which is there, directory is restraints. So these are restraints to guide the dogging process. So these are files that we are, again, the generation of those files is described in the tutorials, but they're already there in case things will go wrong. We have the runs directory. So the runs contain example of the run, of the output of HADDOCK, basically. So these have been pre-calculated so that we can move to the analysis. And then we have the scripts. So here we have some scripts that are used to extract some information from the runs, so that for easy analysis. And then there is the workflows. The workflows contain actually the HADDOCK workflows to run the recipes. And then, so this is the script. Oops, sorry. I'll go away. So this is the script, basically, until this, which is written to submit on Discoverer to the batch system Slurm. So we see we ask for one node, give the account. This is the school-specific accounts where to run, because you need credential, you need computing hours accessible. We give a name. In this example, we're going to ask 50 cores. So we're going to run HADDOCK on one node requesting 50 cores. So we're going to run 50 computation in parallel. We load the modules that are required to activate the software, and then we call HADDOCK3 using the recipe which is already provided. Okay, so I will just submit this one. Before we could submit this one, in principle, you would have to do all kind of pre-processing, but just to check that. So now it should be there. And you see, no alias QU is an alias I defined for such a one. So it's SQ, which is a Slurm command. Okay, that's just my own alias, but basically it's checking my runs, and I have two runs in the system. One is the interactive shell to the node to be able to do the work, and you see this HADDOCK3 run, which has started on node FN2. So things are running. If everything is correct, now you see that there is a new directory that has just been created, which is called run1. So this is created by HADDOCK now. So HADDOCK is running and is going to do calculation. So for now, we just let it run, and we'll look at the results later. Okay, so back to the tutorial. So we're going to prepare the PDB files for the docking purpose. So this is what we need to do. Okay, so we're going to get the PDB files from the PDB database. It's called, we're using the PDB fetch command. We're going to clean it a little bit. Then we want to select a specific chain. Of course, you need to know beforehand what to select. So if you're going to go to the PDB, you should look what is in there. We remove heteroatoms, so we want to get rid of crystal waters. In this case, there is nothing we want to keep. You know, if you have a protein with a ATP in it, maybe you want to keep the ATP because it's important for the binding. So that you have to know what you are doing, but it's important to look at this. Something specific about antibodies, the numbering is sometimes strange in the hypervariable loops because you see a numbering where you will see insertion. So you will see residue 100, and then if you look in a PDB, you see 100A, 100B, 100C. Okay. If you don't do anything about that, many software will see 4 times residue 100 and will only use one of those. And the other 3 residue will be removed. That's a specific thing for antibodies. So you have to check what's happening. And we have a command to fix this insertion. Basically, it's going to renumber things downstream from 100. So it's very easy. You know, how software deal with that depends on the software. In our case, we need to renumber those. So this is what this fix insertion does. Another command that we have here is this PDB select at alternate location. If you use crystal structures, especially if the resolution is very good, high resolution crystal structure, what you might have in a PDB file is that you see 2 conformations of a side chain, for example. So it means that in a crystal, they see 2 conformations. So the side chain is here and the side chain is in another direction. And the occupancy of those might be 50-50. So it means 50% of the conformation are in one, 50% in the other. Now, for doing modeling with those, an amino acid does not have 2 side chains. It only has 1. Okay, so you have to decide what to do with those. And this tool, PDB cell at location, is going to select by default the most occupied one or the first one. If you don't do that, then your software will return an error, but you say, oh, I have 2 copies of the same side chain, I don't know what to do with it. Okay, GROMACS will probably have the same issues. If software takes 2 copies and they do computations with that, then something is wrong, because physically it's not possible. So this is basically what this is doing. So you see the first command here. So this is now going to copy this command. So this is getting entry 4G6K from the PDB, doing some cleaning, selecting chain H, the heavy chain of the antibody, deleting the atom, fixing the insertion, fixing the multiple conformations, selecting only a subset of the residue. For docking purposes, we just want to make the molecule small so that it's faster. And it's writing that to chain H. And the next command is going to do the same, but for the light chain. Okay. So where is my shell? So here. First, I should, of course, load the module. Loads Python 12. Okay, no error. If I list what I have, I've created this file. Okay. And if you look at this file, now it contains only the coordinates. Starting at 1, the chain number is H. And if you look at what is at the bottom, it finishes with H. Okay, 1 to 20. So now I do the same for the light chain. Now there's a new file which is called L. If you look at what's in this one, you see L chain, which also start at 1. Okay, so you see that we have an overlapping numbering between L and H, and L and H form one molecule. So we'll have to deal with that. All right, so then now we merge those 2 chains. So I merge H and L. I renumber everything starting at 1, so that I have a consistent numbering, and I call it chain A. So this will be my first molecule for HADDOCK. And now I have a file which is called clean. And this, if everything goes well, should be exactly the same file as is provided in a PDB file, PDB directory, and you see there are no differences. Okay, so The PDB has not changed since we did that. So now we have our antibody, which is in principle ready to go. HADDOCK compatible, no overlap. Now we look at the second molecule, which is the antigen, sorry, which is interleukin 1b. And we're using the free-form structure of that protein. So not the one from the complex, but it's a free-form structure. So here, same thing. So you see now that I've generated these files. Okay, so now I have both files ready to go. So next step is to define the restraints. So restraints for CNS have a given format where basically we have, so this is CNS format, explore, if you know so those tools. So we assign, and then you have two selections, and we define a distance, a lower bound correction, and an upper bound correction. And the selection, so CNS is very powerful in what you can define here. So here you have one example of such a restraint. So this will be, for example, a distance restraint between the beta carbon of residue 10 and 200 in chain A and B. This could be a cross-link restraint from a mass spectrometry. So we say assign seg ID A, so chain A, residue number 10 and name C beta, chain B, residue number 200 and C beta, and then you see three numbers. So 20, the lower bound correction is 10, the upper bound correction is 0. So it means that the range which is allowed for this distance is 20 minus 10. That's 10 angstrom minimum. And 20 plus 0 is 20. So the distance range in this case is between 10 and 20 angstrom. Okay, if the distance is within that range, the energy will be 0. So that's the way that, so we can fine-tune restraints. Now, for the antibody, you have to identify those loops. This information can be found in a SAP-DB, or there are tools to do that. We wrote, for example, an AI model to predict from sequence what are the binding regions. And this is what we are using in this tutorial. So we're not going to run it. It's not running as a web portal anymore because of security issues, but we have the list of amino acids that form the binding site here. So we can look at those on the 3D structure just to visualize. So I'm going to do that, but I'm going to do that on my laptop. So if I go, so I have here the data of the tutorial, but on my laptop, so we're going to look at the, so this file, this is the file that we basically just created. I'm going to open it in PyMol. So you see this is not the full antibody, this is just the regions that contain the binding sites. And we're going to visualize the binding region. So if you're not familiar with PyMol, you can copy paste those commands. And in PyMol, there are two regions where you can paste commands. Well, there's one region where you can paste, at least for the Mac version. And down there, you can type commands. But here you can paste. And now you see that we have here in red. So these are the loops that have been predicted from this AI model to be part of the binding site. So that's the information that we're going to give to HADDOCK, okay, to define restraints for the antibody. And here, if you talk about show surface, this will be the surface representation. So you see on the protein, this defines, this is a rather well-defined binding site in this case. It's not like it's patchy and you have multiple sites. It's well-defined. Now for the antigen, interleukin 1b, we are looking at this 2013 paper, so it's an old story. So actually AlphaFold has seen this complex, so it should be able to predict it. That's one of the bonus questions if you want to look at it. And in this paper, there is this table where they look at two antibodies, but this is the one we are interested in. And they have done these experiments, NMR titration experiments. And from the NMR data, they identify which are the residue that are affected by the binding of the antibody. And these are the green residues here highlighted in this table. So this is the experimental information that we are going to use in HADDOCK to drive the modeling process. So here, same thing, we can visualize this binding site. So now I need to load the antigen, which is 1I for I1B clean. And this is the binding region. So if you look at it, basically from NMR, you see that there's a rather well-defined region which is identified. It doesn't mean that the neighboring residues might not be involved in the binding. Okay, so it could be that NMR only identified a subset, or maybe some of those are not involved in the binding directly, which is why by default we remove 50% of the data. But this is how it looks like. So in this case, it's a rather well-defined patch. And again, what I just showed you is also represented here on the website. Now, here there is an explanation, basically what I also explained you in the lecture, that since experimentally you might not be always sure that you find the complete binding site, and since we're going to drive the modeling using data for HADDOCK, it's important that there's a good coverage of the binding site. So you rather be too generous in defining your binding site than too conservative. For the antibody, it's not a problem because we know the action is in the CDR loops. For the antigen, it might be a problem. So there is a command coming with HADDOCK, which basically, given a list of active residues, is going to return all the surface neighbors of those. The server will do that automatically for you if you are to use the server, but here we need to do that. So I'm back on Discover, still in business, yes. You see, if I give this command, so I give HADDOCK restraint, which is a command line interface to HADDOCK3, define passive from a list of active residue. I give a PDB file, I give a list of active residues. These are the NMR identified residues. And what it's giving me back is a list of surface neighbors. So that's information. So we can, again, visualize everything. This is what it's doing here. So if I go back to my local version, Bimol, now you see the green ones are what has been selected automatically by the tool as being surface neighbors. So basically the binding site that the antibody can target in this case is the union of the red and green ones. Okay, that's what we're going to use. Let me check. My run has finished, by the way. It's no longer in the queue. The only run that you see in the queue for my name is the interactive session that I started first. So if I look at content of run 1, these are all these directories that have been generated by HADDOCK. But we come back to that later. But it did finish, and there's a log file there. Actually, there should also be a Slurm log file you see here. If I look at the content of this file, you see the output of HADDOCK, where it's reading the workflow first. This is the workflow. It's actually validating the workflow. So if there's a module it doesn't know, it will raise an error and say, I don't know what it is. If you define wrong parameters, it's going to raise an error as well, say, I don't know what this parameter is. And then it's starting topology building, rigid body docking. Capri eval modules are basically analysis modules. You can give a reference structure to that if you want to compare to a reference. It means that you know the answer already. So this is what we are doing here. And otherwise, it takes the best model. It's still useful for visualization. Flexible refinement, and then at the end you add clustering, some cleaning at the end, and here you see that, so this run took 6 minutes and 6 seconds on Discover to run. So that's quite fast, but this is a limited sampling that we are doing. So back to the tutorial. So now we have the PDBs are ready. We generated the PDBs. We have a list of active and passive residues. So now we need to generate this restraint format for HADDOCK, okay? And for that, we need to give it 2 files. The first row of that file contains the list of active residues. So these are the ones that we really want to be in the interface. And the second line contains the list of surface neighbors, the passive one. For the antibody, the second line is empty. For the antigen, we have These are the first line are the NMR identified residue. The second line are the surface neighbors. Okay, those files are provided in the restraint directory. So I can generate. So now I give the command from the tutorial. So I go active passive to ambig. So this is the restraints file. I give the list of active passive for the antibody. I give the list for the antigen. I assign chain A to the first one and chain B to the second one. It's important that your restraints file match the chains in your file, otherwise it will not be recognized and discarded. And I create a CNS file, and if we look at what is in there, you see this assign statement, and now you see it looks very weird, but so this is, I say a distance is defined between two selections. So the first selection, we have the first bracket. So it's residue 1 and chain A. So that's a residue of the antibody. And then you see the second selection, the bracket opens here and closes here. And what's in between is the entire list of residue that form the active, the NMR identified binding site plus the surface neighbors. So you see there is this residue 72 and seg ID B OR. So you have this OR combination of the entire binding site. So it's targeting the entire binding site. So it looks like a weird distance restraint, but it's again a distance restraint between many points. And you see that the distance, it's between, so 2 minus 2 is 0. So the lower limit is 0. We have van der Waals interaction, so we don't need to worry about atom overlapping. And the upper limit is 2. Again, because we calculate many distances and the effective distance will always be very small. And you will find such an assign statement for each of the active residue that you define, you will find such an assign. So if I do, so this is basically, so these are all the, I'm just grabbing the line that start with assign here. So these are the, so if I looked at how many are those, so there are 43 lines, so meaning we have 43 distance restraints that we're going to use to drive the modeling process. And randomly, we're going to delete 50% of those for each model that we generate. So each model will come out of the fraction of those restraints. Always going the wrong way. Okay. Okay, I just show you this file. And what is the distance range? I just explained you it's between 0 and 2 angstrom in this case. If you write your own file and you were to make a mistake, you can validate to make sure that there is no problem in there. And here, if it's no output, it means it's okay. Let's make a mistake. Let's delete this parenthesis. This should return an error. I didn't test that, so it's real life test. Okay, so this is Python output, but now it tells me invalid table file. There is a malform assign statement at line 1. Okay, it's pointing you that you have an error. So if you manually generate those files, before using them, you want to validate if it's not validating if the residue exists or the atom exists. It's just validating the syntax. If you open a parenthesis, you should close a parenthesis. So this can't, well, this is because it's very hard to validate that the content of the file is correct, but you can validate the syntax at least. So that's simple too. So let's fix it again. Okay, so now we have here our input files. We have the restraints file. In principle, we have everything that we need, almost everything that we need. There's one more thing that we need to worry about, and this is specific for antibodies, but for other protein, it might play a role as well. So antibodies consist of a heavy and light chain. And those chains are in principle, if we are only modeling the tip of the antibody. So those chains, there is no covalent bonds between those. So just they are held together because of non-bonded interactions, electrostatic and van der Waals. In the flexible refinement that we do in HADDOCK, this simulated annealing protocol, we start at 2000 Kelvin, the molecular dynamics. And only the interface becomes flexible at some point. The remaining of the molecule are rigid, but they can still move around. So because of this high temperature, there is a lot of kinetic energy in the system, and it could be if the chains are not covalently attached to each other, that they might drift apart a little bit, simply because of the energy that we put into the system. This could also happen for other proteins where you have multiple domains, and when you cut a part of the protein, effectively you have two domains, but they are not physically connected anymore. Okay, so in those cases, this is something that the web server does automatically. When you upload a PDB file, we are checking if they are disconnected domain. This could also happen because you take a crystal structure, a loop is missing, and that loop is the connection between two domains. So then physically what you have two bodies that are not covalently attached to each other. So there is a tool which basically checks the PDB file, and if it detects breaks in the chain, it's going to pick randomly a few distances which are imposed during the docking just to keep the chain together. So this is what this tool is doing. So if we look at what is in there now, you see it has selected two distances. They both belong to chain A because this is the antibody part. And it has measured what is the distance in the structure. And it imposed this distance with 0 error bonds. So you just want to maintain this distance to make sure that the chains stay together. Keep going the wrong way. Okay. So now we have all the data. We have the PDB files, we have the restraints files, so we can define a workflow. So the workflow is going to basically mimic a bit the workflow of what the web server is doing. So we're doing a simple workflow in this case, and we add some analysis steps in the workflow. So this is what we're going to do. So we call first topo A, which generates the topologies and builds any missing atoms. Then we do rigid body docking by energy minimization. So that's the first stage in HADDOCK3 series. We add a Capri evaluation module, which does some analysis of the models. And here we're going to give the reference structure that we are going to reproduce just to be able to measure how good are the models. If you don't have the reference structure, you can still give Capri eval if you want to get a plot of the distribution of score versus RMSDs, but then what HADDOCK does is to take the best model ranked by HADDOCK as a reference. And then you compare all the model to that one. But that allows you to make plots of how is the convergence of the modeling. Now, then we select, so here we might generate, in this case, a few hundred only, but if you don't define the sampling, the default will be 1,000 models. Then we select a number of those models to give to the flexible refinement that takes a bit more time. We perform this simulated annealing performs refinement of the interface, again, some analysis module, final energy minimization, analysis module, clustering. That's what this cluster FCC does. It's based on the fraction of common contacts. So it's not an RMSD-based clustering, but it analyzes the contact at the interface between the molecules, and it clusters based on the similarity of contact. So this is much faster than RMSD clustering, and it works nicely for proteins. We select the clusters, we do a Capri evaluation step, and then we add another step, which is a contact map analysis, and this is going to generate plots of the contact across the interface. So we can visualize what is in there. So there is a lot of graphical visualization of the results. Now, this is the docking antibody-antigen workflow, which is provided in the tutorial under the workflows. And this is the one which was used in the Slurm submission script that I used. Okay, so here is the what is in there. So you define a name for your run. So it was run 1, and you've seen that run 1 did appear in my directory there, but you can give it an arbitrary name. What you don't want to use is spaces. If you are a Linux user, you should know that space is not a good idea. And then the mode that we are using here is it's called local, and we request 50 cores. Because what we are doing is to send the entire HADDOCK workflow to a node on Discover, and then we use 50 core on that node. And HADDOCK is basically knowing in that way that I have 50 threads that I can use to calculate the model. So it's going to calculate 50 models in parallel. So it's different. GROMACS will do one calculation using 50 core. So here we do 50 calculation using one core. Okay, so it's the high throughput mode of HADDOCK. There are other modes, so you can also run in MPI mode, so it's message passing. So then you could say, I'm going to do a very large sampling. I want to use 4 nodes of Discover, and each node should use 100 cores. Then you are using more cores than is available on a single node. But that's a different compute model. So these are default options that are turned on by default. So we do some post-processing and cleaning to limit the number of files. So HADDOCK does generate a lot of files, okay, because we write a lot of PDB files, and then we give the input molecules. So this is our pre-processed file. If we were to use an ensemble of conformation, I spoke about ensemble when discussing the cyclic peptide docking. This PDB file could contain multiple models. So you have this model and model construction in PDB files to represent multiple conformations. And then you have the definition of the workflow. So I called topo AA, I called rigid body. I define my restraints from the CDR to the NMR epitopes. I define another set of restraints called unambiguous in this case to keep the antibody chain together. Everything which is defined under those unambiguous names is always used. Things that are defined under the ambiguous names, by default, 50% is removed. You can turn off that option. There is an option to turn off that. It's one of the parameters. And instead of doing the default of 1000 in this case, just for speed purposes for the tutorial, we're only going to generate 100 model. Effectively, it means that the sampling is 1000, because for each model that we write to disk, internally, we sample 10. So we do 10 trials. Actually, we do 5 trials. We get a model. We systematically rotate 180 across the interface and re-minimize that model, because we realized over the years that often we get symmetrical solutions. So if you have a binding mode, which is a groove, you could think I could fit in this way or this way. And sometimes it's not easy to distinguish those two binding modes. Again, our distance are not telling us in which orientation it should bind. All orientations are possible. So internally, we do a sampling of 10, but we write the best model to disk. So when you say sampling is 100, effectively we have sampled 1000, but we write only 100. Then we do this analysis step. And in this case, since we know the answer, we provide the answer as a reference, so that when we look at the statistics, it will be directly calculated compared to the reference, so we can see how good the models are. In a real case, you will not have a reference. So in a real case, you could just give Capri eval without reference, and then it use the best model. We select the best 50 models based on HADDOCK scoring function. And then we go to the flexible refinement stage, where we optimize the interface. Again, we define the 2 restraints file that we needed. We do an analysis step, and we do the final energy minimization step of HADDOCK, again with the restraints. Analysis, clustering, selection. So here we select 4 models per cluster, analysis, contact map. Okay. So this is basically what has been running. So this run in about 6 minutes in this case. Typically, if you are smart and you want to maximize the use of the resource, you want to have the sampling, which is a multiple of the number of cores that you are using. Okay, if you were to do a sampling of 200 with 50 cores, then you will go 4 loops of 50 will give you 200. If you do a sampling of 201 with 50 cores, so you do 4 loops of 200, and then 1 with only 1 model and 49 cores that are doing nothing. So in terms of using the resources, it's not very smart. Okay. Now, there is a lot of parameters that you can define, and the workflow that we are defining are very simple, but they are very advanced parameters. So if you want to know what are all the parameters that are associated with the module, you can use these commands. With m is the module name, and then you get a long list with a very short description of what the options are. Like if you want to turn off the random removal, you could figure out which parameter should I need to that, and then you add it to your workflow. So this run was about, on Discover, about 6 minutes using 50 cores. On this laptop, it takes about 7 to 8 minutes using 12 cores. So it's also quite fast. Okay, so I explained you about the local mode. I told you something about MPI. This file I already showed to you. So this is the file that you have if you copy, if you get the zip archive, which is on Discover itself. If you download from in the instruction running on your own, you can download the archive from the web. It does not contain this Slurm file. Okay, so then if you're using a different system, you will have to write these scripts. And this was the way that I submitted. And these are other systems. So recently we gave a workshop in Istanbul. These are instructions to run in Istanbul. This is also a Slurm-based system. So it looks very similar. You have the same. So this is Slurm-based syntax, but we use here, we have different times. It's just, and earlier this year, we were in Singapore, and there we were running on Fugaku, which is a large supercomputer in Japan. And then they have another type of system, of batch system. Anyway, it's not important for here. Okay, so now we have run everything. I've run everything on Discover. The runs are there. And this is what you see as listing of run 1. Okay, so we have the different steps. So each step in a workflow has a number. So we number sequentially the steps. So we have 0 topo A, rigid body, up to 11 contact map, and then is analysis, data, log file. So if I look at now which direction I go, yes. This is the content of run 1, which I just run on Discover. You see it goes from 0 to 11 contact map, analysis, data, log, analysis, data, log. So it's exactly the same. Okay. So for each of those stages, there will be files generated. So let's go into run 1. What is in 00 topo AA? You see that those files have been zipped. That's the clean option. So the clean option, which is by default true, is compressing the file at the end to save space. But you see here, so the input PDB files, and these are input PDB files that have been processed by HADDOCK. So if there were missing atoms here, the atoms will be part of that. So let's, for example, just show you. So this is, okay, it detected 2 disulfide bridges. So this is also automatic. So detection of disulfide bridge is automatic. It detected that there was no peptide link between residue 120 and 121. And these are the 2 chains of the antibody. So they should not peptide bonds. So it's based on distance. And then you see the residues. So it starts with GLNN. And you see, for example, so these are, if you look at it, you see that not all atoms are present. For example, the beta carbon only has no hydrogen attached to it. So by default, HADDOCK uses a kind of a united atom model. So we, by default, delete all the hydrogens that have no partial charge. And this is to speed up the computations. And this saves about 50% of the number of atoms. Okay, so your system is smaller, it's going to run faster. It doesn't affect really the quality of the model. Because it's a united atom model, but you want to keep the hydrogens that have partial charges because these are important for hydrogen bonding electrostatic. But a methyl group, you can get away without the hydrogens. So there's an option to keep them all. It's just going to take more time, but it's not going to give you better docking results in principle. For small molecule docking, maybe you want to keep everything. But these atoms, for example, the He21 and 22, we can compare with, so this is the antibody. So this is the file that the 4G6K clean is the file that we just created using PDB tools. And you see the first residue is here, but there is no hydrogen in that one. Okay, this was a crystal structure which has no hydrogen atoms. And you see that HADDOCK has generated the missing atoms, basically. So again, if the side chain was not complete, in principle, it only needs one atom, and then it's able to regenerate everything. And then the other file which is there is this PSF file. So if you are coming from CHARM or NAMD, then you will know this is the file that defines the topology of the system. If we look into the rigid body module, we ask for 100 models. The 100 models are there. Okay. Looking at this, you might not know which model was the best one, the better one here. But if we go into the 02 Capri-Eval, so that was the third module. So we have Topo A, rigid body. Capri-Eval is an analysis model. And here you have this Capri. So this is a tab separated value. So it's a file you could open in Excel if you like Excel, but it's a simple text file. Look at what's in there. So here you get basically the statistics and the rankings of your model. So this is telling you that at rank 1, so this is the rank, model 42 is the best scoring one. You get the HADDOCK score here, and then you get some statistics of the quality of the model. Interface RMSD, fraction of contact. So this one is not very good yet. It's 5 angstrom away. If you look down, there is one here which is 1.2 angstrom away. So the best ranked model here is not the best one in this case. And you get component of the score. So you also find here, so the first line gives you what is listed. So you will find electrostatic energy, van der Waals energies, and all of that. And also the restraint energy and the dissolution energy. So you can find those statistics for the entire system. So this is the ranking, basically. If you look at 03, select top. So this is not just selecting the best model. And the way the module communicates with one another, because there is information from one module going to the next one, is through this JSON file that contains all the data which is generated automatically. So if I look at 06 EMREF, now you find 50 EM refined model, which is the last stage of HADDOCK. Okay, so all the data are there, basically. So I was showing you this Capri file, which is also shown here in a more readable way. Okay, so now we are looking at the Capri-Eval number 10. So this is after clustering, the best model. You see here the score that we are getting, about 140, with RMSDs that are now below 1 angstrom. And the best model is actually one of the best scoring models. So that's, so what are the statistics that are relevant? So if you know the answers you want to compare to the real structure, of course, you have different statistics. So this is the RMSD at the interface of the complex, the fraction of contact that are correctly predicted, the ligand RMSD. So we fit on the receptor and we calculate the RMSD on the ligand. So this is increasing the RMSD. And DOQ is a continuous score between 0 and 1. And these are all statistics that are used in people predicting complexes. If you have heard of Capri, which is a blind experiment for predicting complexes, it's the equivalent of CASP. We measure the quality of the models in that way. And you find the other terms. So like those terms are the terms that are entering the scoring function of HADDOCK. So electrostatic energy, intermolecular, van der Waals dissolution. Okay, so if I go look at, sorry, 10 Capri-Eval, that's the last analysis module. So I can look at this file. So it tells you, okay, what was the best model? So best model, this is for clustering. So you see the name is cluster 1. So the score in this case is -135 and the RMSD is 0.98. This is what I just got on Discover. And what you see here is is here. It's slightly different. This is what I got by running on my laptop. So you have to realize that you will never get exactly the same results if you don't run exactly on the same hardware. Okay, CPUs are different. We are adding a lot of real numbers, so it's our floating point operations. So there will be differences between different hardware. Okay, if you run exactly on the same hardware, you should get exactly the same results. But if you run on the different hardware, there might be differences. You don't expect that the difference will be too large, but there will be differences. So hopefully, the best models still are very close to the best model that you get somewhere else, but it's just a bit the chaotic nature of everything which is MD, molecular dynamics related and computing. So how are we doing? So we still have 20 minutes. So this is the single structure analysis, and in the same directory, so this is a lot of data. There's another file, so this SS is for single structure, CLT is for clusters. So this file looks rather similar, but you see, so we had 50 models that we generated at the end, and they cluster into 4 clusters. Okay, so those 50 models are only 4 clusters. So we like to look at cluster statistics because it's much simpler. It limits the number of models that you have to look. And now you see cluster statistics and also a standard deviation. Okay, this standard deviation is calculated. So the statistics is the average over the top 4 models by default. And the standard deviation is calculated also from the average, from the score of the top 4 model. And then you get also then the quality RMSD plus standard deviation. So you see so model 1 is at a 1.1 interface RMSD. The second one is at 5. The third cluster is at 14, so that's a very different solution. And the last one is here. It's at 10. Okay, the columns are not aligned here. So actually the ranking works very well because the best model is put on top. But that we can only say because we know the answer. So now you can look at a lot of numbers, but the good thing is that in the analysis file, so HADDOCK directly gives you an HTML report of your docking run, which makes analysis much easier. If you were to use the HADDOCK server, this will be generated by the server. But in HADDOCK3, we have those files that are present there. So let's go look. So now I want to visualize those. So I'm going to go to a run on my laptop. Run 1. Okay, run 1, we have the same. We have this analysis here. And for each Capri step that you put in your workflow, there will be a directory which allows you to visualize the docking result. So for example, 02 was the analysis after the rigid body. So here we are looking at the 100 models. What do we have in there? A lot of HTML files. So these are statistics and plots. But there's also the report that contains everything, basically. So I can open this one. So this is now what you get. So you can open it with any web browser, basically, where you get now so these are not cluster statistics. We didn't do clustering here, but these are single structures. So you have the rank of the structure. The report is only showing you the top 10, not to have a page which has 10,000 entries. And you get the scores and the component of the scores. And if you scroll down, then you start seeing the models. And in this case, the reference, because we know the answer, is the crystal structure of the complex. So you can see, how are the there is no clustering, but you can already see that this is probably one cluster. These are interactive plots. So if you hover over a point, it's going to tell you what is the RMSD, which is the X coordinate, what is the score, which is the Y coordinate, which model it is, and what is its rank. So you can, and you can zoom in on the plot and select the subsection. So now this is, so if I just want to, if I'm going to select, I just want to select these guys here to concentrate on what's happening here. Come on. Nothing is happening. Or it's thinking about it. It should zoom in. Maybe my browser has an issue, but in principle, it is interactive, and every plot on the row will be, let's see if it gives a different story in Chrome. I want to zoom in, so let's see. Is this working? No, it's not working. Feature. Anyway, you can get an idea of the, how the run looks like. So if you look at this particular plot here, you can see that the score is not perfect here. So there are, different solutions that are showing up. But this is rigid body docking. So we can do the same, for example, at stage 10 was after clustering. So now you get the same report. Let me make it a bit larger, where you get the rank of the cluster, the size of the cluster. In this case, it's 4 because we asked maximum 4. And you get the scores. And you see, for example, from the HADDOCK score that the first cluster is clearly better than all the others because the standard deviations are not overlapping. You might have cases where you have several clusters that overlap in standard deviation, and then you cannot make a selection. You cannot say that one is better than the other. You can change the order. So these are, if I want to reorder from small to large or large to large. So for each energy term, these are interactive plots. And now you see the cluster that are color-coded, 4 clusters. And now you see, for example, this cluster now is scoring way better than all the others. So this is, and these are interactive plots. So if I click here, in principle, they should disappear from the plots. And this is happening. Okay, so there is interactivity. So you zoom in the cluster. So you can have a quick visualization of your results in that way. The first row gives you the HADDOCK score versus, and the component of the score versus the interface RMSD versus DocQ, ligand RMSD, interface ligand, fraction of contact. So this is the similarity of contact. And if you scroll down, you get a distribution of scores. So you get this box plot where you see the distribution with the median, first quartile, second quartile, all of that. So it's nice to see. You see cluster 1 is the best in HADDOCK score, but it's not because of the van der Waals. It's actually because of the electrostatic that it's winning. Because like cluster 3 has a better van der Waals interaction. So this is a nice visualization of your results. So this is what is explained here. Okay. So how many clusters? In this case, 4. The best one was better than all the others. It was significantly different. We look at those plots. So now we provide for this tutorial a simple script that extracts data from this Capri evaluation file just to get some statistics. So I'm going to run that. Let's go back to Discover. Okay, I have my run 1 which completed. So I'm going to call that script. Oops. So what this script is doing is extracting statistics for each Capri stage. So you see here for the 0-2 was rigid body. So we generated 100 models. 19 of those were of acceptable quality, meaning better than 4 angstrom RMSD. 11 were medium, and there was 1 model out of 100, which was below 1 angstrom after the initial docking stage. And here you see some statistics. Whoops. So this is telling you, so this only makes sense, again, if we have the reference structure, but just to give you an idea of what's happening. So you see the first model in this run is actually ranking nicely at rank 1, and these are the values. This is the rigid body. This is after we select the best 50. So, okay, we've lost a few models, but still, if you do a random selection, you will expect that you only select at best 10. So we do the scoring function does work. But you see at this stage, the ranking is not fantastic. This is rigid body. It's only after flexible refinement and minimization that the rank are really working well. So the flexible refinement not only improves the model, we go from, but it improves the ranking. So this is at the end. So the final stage after clustering, you know, we are 4 out of 16 because we have 4 clusters. So there's 1 cluster. The best model has an interface RMSD below 1 angstrom, and it generates about 0.90% of the contact are correct. Oops. We can do the same. There is a script that extracts the same statistics, but for clusters. Of course, we only did clustering at the end, so you can also say, okay, how many clusters? And again, you get these values. So in this example, our best score cluster is actually the best solution. And actually, on Discover, we get slightly better results because our values aren't the same, well, slightly better. Okay. Now, contact analysis, if you want to get you can, of course, you should always visualize model. It's important, okay? So take the time to look at the models. But now since we added this contact module, there is information about contact directly in the run. Seems that I've been drawing in Zoom, I need to, well, there are these weird lines here, but we can live with that. So I'm going back to my run. And I'm going to go back to the contact map module. Again, you see here tab separated files. So this is going to, and for each cluster, there is such a file. Okay, so it's doing this analysis per cluster. So if you want to compare the contact between different clusters, you can open these different files. There is also files that are giving you the contact. So this is going to list the contact. So if I look at this file, you get the listing of contacts and the probability. There is a kind of a probability. It says which fraction of the model is this contact seen. But if I look at contact map reports, or let's look at, so here you get this simple, so cluster 1 to 4. So let's look at so this is a plot which is generated automatically, a little bit larger. So you have the 2 chains. So this is chain A in blue, chain B, interactive plots. And then here you have all the amino acids that are making contact, and they are color coded based on the type of amino acid. Like if you look at the red ones are negatively charged amino acid. The green ones will be polar one. The blue ones are positively charged amino acids. So, and if you go over them, it tells you. And then if you look at this one, so this is lysine 94, and you see that lysine 94 makes 6 contacts from the cord, and it's telling you to which contact, okay? And it's telling you the type. So you can kind of visualize the contact and compare different ones. So that's one of the plots which is provided. There is this heat map, which is also provided, which is this, you might be familiar from other type of software where you see not the complete sequence here. So this is the antibody part. This is the antigen part. So here, this is the contact inside the system. And these are the intermolecular contacts that are made. And again, this is interactive. So if you go somewhere, it's going to tell you. So if I zoom in, so this is a contact between the tyrosine on the antibody and the phenylalanine on the antigen. And the color coding is the probability, which is mean in how many models within cluster do you see that. So is this a contact which is always present, or it's only present in a small fraction? So it's just for analysis. This is interesting to look at. So this was the contact map, and maybe another one is traced back which is, I think, not listed in the tutorial, but if we open that one, just to show you what's in there, make it a bit smaller. So now for each, basically, so this has been run, this is run basically on the final set of clustered model, and it's telling you, it's giving you the sum of rank of that model throughout the different stages of the system. So it's, what was the rank of that model in a rigid body after flexible refinement and so on. So if a model which is very good, you will expect that it's scoring very well at all stages. And you see, for example, this model, the sum of rank is around 30. And the different colors give you what was the rank at different stages. So like this one is a model that was scoring pretty bad at all stages. So this is also, a model that scores well at all stages gives you more confidence that maybe it makes sense. It's a good model when you do, because in a blind case, you don't know the answer. So this is another useful, and it also tells you, which model was it and what was the rank at different stages. So you can compare how the rank is changing as function of the stage that you are in. Okay, so wait. So this was the contact analysis. So now we should take a look at those models at the end, because visualization is critical to everything that you are doing. And the models, the final models are in this directory. So we're going to look at the models in PyMol and compare them directly to the reference. So this is the complex that we want to reproduce. Okay, so I'm going to go to 09. And here you see that there are models for there are 4 models per cluster. So I'm going to load in PyMol the best model of each cluster so that we can compare them. And I'm going to load also the reference structure, which is known because we in this case, we have a crystal structure. So I'm going to ask PyMol to load everything which is 1.pdb.gz. So PyMol can, by the way, load gzipped files, so you don't need to unzip those to visualize those, so it does it on the fly. So this command is going to load the first model of each cluster. And on top of that, I want to load the file which is the reference XM matched. Okay, so now I have the 4 models, 1 model per cluster, which is the best one, and the crystal structure, which is the matched one. So doing some visualization. So now the crystal structure is in yellow. Our models are in blue and green. So blue and green are the 2 chains of the antibody, but we have all of those. And we want to align basically everything to the reference so that we can compare things. So I'm going to use an align to 4G6M match and chain A. A is the antibody, so I align everything on the chain A, the antibody chain of the reference. Now that looks better. Okay. And now we can see the different models. So this is the top rank model by HADDOCK. We started the modeling from the apo form of the protein, so it's not the complex. So you see that there are still differences at the interface if you look at the backbone, but in general, the model that we did using only interface information in this case is quite good. If you look at the other solution, so model 2, you see it's very different. So can you see the difference between model 1 and model 2? It's hard to see, but so this is model 1, which is the correct solution. This is model 2. Look at these headings here. In one model, it's here. In one model, it's there. So those 2 solutions are 180-degree related. So basically, it's a 180-degree rotation. So it puts, again, we don't define specific contact. So if you think of two surfaces interacting by rotation, they might still form they might still fulfill the information that we have. Of course, the score makes this one the preferred solution, and this is the correct one. And this was the second best scoring solution, but it's a symmetrical related solution. The next one is very different. So that one is shifted actually. And the last one, the worst scoring model, is again a very different solution. Actually, it's also a 180-degree rotation, that one, but a slightly different one. So now let's visualize the data that we put in, basically the binding site. So now this command basically has been colored in red and orange the residue that we gave to HADDOCK to define the restraints. So the paratope on the antibody and the epitope defined from the NMR data. And you see that these are the interface, but that's not surprising because this is information that we give to HADDOCK. Of course, the contact are going to be through the side chain. So, if you look at there and you zoom what's happening here, you see that all those residue that we defined that should make contact are actually making contact. But this is what HADDOCK is meant to, just to force the residue to make contact. But remember that 50% of the data were randomly removed when we do this. So we're not using the full information, but we randomly sample out of the 50%. And this is basically what I've just been showing you. Okay. So, well, you could not run things on your own. I could run it on Discover, and you've seen it's working. Yes, and I got my results in 6 minutes for this limited sampling. If you were to do a full sampling, probably you will get your results in about 30, 45 minutes for one docking run in this case. So it's still using 50 cores, so that's nice. Okay, so there's still 15 minutes. So, okay, so we can do bonuses. Good. Maybe first, since, you could not be active, you had to follow, you were passive. So first of all, are there questions? Yes. So this is for any type of proteins, it will work in principle, yes. Yeah. So if you process the one where we're processing the PDB files, you mean? Yes, so you can do for any type, but of course you have to check what are the chains in your file. Because here we are processing an antibody, and antibodies, the convention in a PDB is that it's H and L. So before processing a file, you should go to the PDB and look at what is in that file actually. There is actually a command in PDB tools that should, we had done, I think it was PDB fetch that we are doing, and what was it? History, a lot of commands that we've been doing. So this one. So you can go to the PDB site and look, what is in there. Often, maybe you can realize that in the PDB file, there are multiple copies of the same molecule. So you want to select one and not everything. I think this PDB work count, if I'm correct. Yes, so now I've used this command, PDB fetch, and I've pasted it to a command which is called PDB_WC for the word count for Linux. And it's telling you what is in this file. So here, let me just bring it up a little bit. So the output of this command is telling you, okay, there's one model that consists of 2 chains, number of residue, number of atoms, number of heteroatoms. In this case, there were no multiple occupancies. So no, we did this check for multiple occupancies, but it will not have been needed. And there were not there were no residue insertions. So this is an example where you don't have this weird numbering of the loops. If we do the same for the antigen, just to get an idea, so what was the antigen for I1b? 1b. So again, this is now, you see, there's one model, one chain. It doesn't tell you what is the chain, so you will still need to know what is the chain if you want to select, but in principle, yes. So anything which is peptides will work. Nucleic acid, RNA should also work. That will be because you need to have the force field defined. So there's a way. Do we have? Let me go to. So I have a version of HADDOCK3 which I install locally. So there's a version with module, you use the global version, but first I install it locally. And if you do a clone of the Git repository, it comes with examples. So I'm going to show you one example of protein small molecule workflow. So these are all examples of workflows. Protein ligand. This is a small workflow, this one. So if we look at what is in there, you see here for the topology, now I'm giving for the ligand a parameter file and a topology file. So this contains the specific parameters. So I'm not providing the PSF. These files are, so if you are Charm user, let me show you what is in one of those. Just if you are a Charm user, it looks a bit, so this is the topology file where you define the bond, the angle. So it should resemble Charm, but it might not be exactly the same. So that's, and then there is, well, there is not really easy software to, and this is the force field. So these are, you have the non-bonded. So this is probably Charm. You have the 1,4 that are scaled down, so all of that. And then you have the definition of, actually the bonds are not, the bonds and the angles are here. So here you have the definition of bond angles. But it's important if you provide your own file to make sure that those naming here are not overlapping with the naming of the protein and the other force field which are there. So it's force field are tricky things. We hope to have in the future a way of parameterizing more easily small molecule that will, so AC pipes, you can do it. BioBB has a workflow to do that, but it's, and you have to test basically, the test, you have to do an energy calculation to see if your force field, because, if, until you go to the energy calculation, you don't realize that you are missing parameter for an improper or things like that. So that's, so that's fine. That will work. Yeah, nucleic. Yeah, that's so nucleic acids, DNA, RNA. So the difference is in the naming. So if you have the name of residues in DNA will be DA, DC, DT for deoxy, and that's a PDB convention. And RNA will be ACTU. So this is how it distinguishes things. So you could probably even do a mixed DNA RNA molecule. So that should work out of the box. Glycans, we support a number of glycans. So there is also an example for glycans. You see this peptide cyclization. I told you we can do in one go this cyclization protocol. So it's there. So that's more advanced. There's a glycan one. If you want to know which molecule are supported, then we should go to the website of HADDOCK. So here we have online a list of molecules supported, list of modified amino acids and other molecule types supported by HADDOCK. And here you find all the carbohydrates. So these are the ones that we know. Carbohydrates are tricky, but because there's a lot of different linkages that are possible. So, and there are a lot of different carbohydrates, but this is the one that are working. There's no guarantee that all the linkages are there. These are the ions that are supported. There were people asking about docking ions. That might be tricky, but the ions need to be written in a format that specifies the charge state. Because in a PDB file, you will only find zinc, but some ions have multiple charge state possible. Iron is an example. So for knowing which parameter to use, you have to have the charge state. So we have phosphates, sulfates, heme groups, DNA, RNA, and then we have here the list of modified amino acids that are present. And So if these are there, then you can use those ones for, and the way to make a modification will be simply to edit the PDB file and change the name of the amino acid. You don't even need to bother to put like if you want a phosphorylated histidine, just edit the file and HADDOCK will generate it for you. So I don't know if there is a histidine in the, so if we go back to my own discoverer, yes, I'm here. Well, projects, HADDOCK, antibody-antigen. Is there a histidine? Let's see. So we could make a mutation just to let's do we have primol there? Yes, I have primol. Is there a residue? Here is a no, that's a proline. Tyrosine. Well, there's a tyrosine here, for example. No, this is a tyrosine. So I want to, so this is tyrosine 152. So if this one was phosphorylated and I wanted to calculate, to use it in HADDOCK, I will edit the PDB file, change the name of tyrosine 152 to the corresponding name. So I have to find what is the name of a phosphorylated tyrosine. Phosphotyrosine is PTR. Okay, so if I change the name to PTR, it's going to generate the phosphate automatically. We can do that. I'm going to do that on my laptop. Then we can visualize. Data. Yeah, so that there will be, if you do a mutation, typically what you want to do is a minimization or maybe a flexible refinement. So if you want to, so you could use the module, which is, so we have an EM-ref module, we have an MD-ref, which will put water and do a bit more refinement. So you're not doing docking, you're taking and just refining. And this might be able to remove clashes. If they are really bad clashes, the system might explode. Energy minimization is quite stable, so you can always minimize. Molecular dynamics, that's where the explosion takes. Yeah. So you want to put a minimization first and then. And it won't support the minimization, it's going to pull them apart. Yeah. So for example, we have, so we say, so this was our reference structure, okay? So we say it was tyrosine 152. No. Okay, so this is 152. I change it to PTR. I don't delete the atoms that are not needed. Well, in this case, it's adding something, but if you want to make another mutation, just don't bother deleting atoms. Because like, for example, the C-beta atoms give you the direction in which the side chain should be rebuilt. If you change from an arginine to a leucine, the beta information is not. So it's going to reuse the atoms that are common. Test.pdb. So I wrote a new, I don't want so. So one command that I didn't show you, we still have 2 minutes, is HADDOCK score. So HADDOCK score is a command line interface where you can give it a complex and it's going to calculate the HADDOCK score. What it's doing is a very short minimization and then outputting the score. So if I say 4G6M matched, so this is now on my laptop, minus 147. Now I do the same for the test. Let's see. Now that's a live. So now it should build first the phosphate, and it should give well, it's even better. Okay, so we improve the binding, but okay, you know, that's I'm not sure, but well, you know, we can play this game, so we can kind of try to do design. So there's also an alanine scanning module, which we systematically scan all the interface residue to alanine and give you the change in delta score. So you can identify which amino acids are having the largest energetic contribution to the binding and which the worst. But you could also scan to some other amino acid. So I want to run output PDB. So it's doing the same, but now it has written a file which is the minimized file with the mutation. So now we can go check if the phosphate has been added. Okay, so that's a real test. Again, not prepared, so let's see if it works. Full trust in the software, of course. So the question is, so let's align all to this. Okay. And you deal.cbc, yes. Zoom, res ID 152 and chain A. So sticks. And let's color everything by elements. So where is
the phosphate level?
the phosphate is not, no. Is it 152? Tyrosine 152. Where, oh, show sticks.
Yep.
The phosphate is there. Okay. So it rebuilds. So you can do a mutation, you can play this mutation game like that. So, you know, you can change names, or you can do the systematic scanning, or the alanine scanning can allows you to define, I want to mutate this residue, and you can give it a list of mutations also. And it gives you a plot, HTML interface, interactive HTML plot where you can look at the results. But so in this case, it minimizes it, it put it there. You know, it doesn't look crazy. So there is a hydrogen bond with another tyrosine here. There is a lysine here. This is an asparagine, I guess. So it put it somewhere that makes sense. We didn't introduce clashes. The score is better. Will it really survive if you were to do the experiment? I'm not convinced, but maybe we are lucky. Maybe it's a lucky shot. Probably if you had a second phosphate here, there is another tyrosine here. Mm-hmm. That should not be very good, I would say, because then you have two negatives. So, you know, that's so this is 178. Yeah. So yes. Beginners. So we have a lot of tutorials. So take time to run those tutorials. Some of them you can run, you know, on your own system. If you are really beginner and you're just interested in doing some modeling, HADDOCK3 is all command line. So you can do a lot of things, but for basic stuff, the server might be the easiest way to start with, to get familiar with things. And then depending on the computational resource, then HADDOCK3, you have more control. You can do different things. Like this scoring option is not part of HADDOCK2, so this can be very useful. So we have a lot of tutorials for different scenarios, basically.
And for HADDOCK3, is there a way we can use the same bit as HADDOCK2?
The server, no, there is no, no. Because that's also using the grid is, so you need a passport for the grid. So it's kind of, we're using these X509 certificates, and you need to be, and HADDOCK3 doesn't have yet a running mode that will be able to submit to the grid. So we have MPI mode, we have a local mode, which is what we use, and we have what we call the batch mode, where you will be starting HADDOCK interactively, for example, and it will be sending to Slurm. But then it sends a lot of small jobs to Slurm. So the HPC system administrator will not be happy with you if you run HADDOCK3 in that way. Okay, so that's not the best way to run. But we have not yet a grid mode. So I think we are working on a web server for HADDOCK3, and most likely we want to use more cloud computing resources there, where you say, give me a cloud machine, and then you start one full computations on the cloud machine, and then retrieve the results. Because now the computation has started on our server, and HADDOCK is sending job to the grid, and then jobs are coming back. And for HADDOCK3, the mode will be, we send the entire computations to the cloud and get back the results. Yeah. You can, in principle, access grid resource. You have to become member of a virtual organization. You can get a 509, X509 certificates, and then you can start using it. But you have to learn the dirty tricks of grid computing, which are not so simple. Yeah.
I've run out of time. Let me thank first Alexander that he covered in a great way the problem with Discoverer. Thank you, Alexander.
Yeah, you're
I
welcome.
hope it was still useful for you. You learned a lot of things from the source of docking with HADDOCK.